leaf and it's infected, okay, and you see these infected areas of the leaf that are going to actually start dying, okay. But people found that, in fact, again, this was transmissible. They could take the sap from an infected leaf and they could rub it onto another leaf and it would become infected, okay. But what was surprising is that they could pass it through a filter that was extremely fine, that at that point when they did these experiments had been thought to filter out any bacteria. Okay? Yet the infection still occurred. So what, of course, initially what they thought, well, there's some sort of organism that they hadn't known about before that's even smaller, much smaller than bacteria. Okay? And those turn out to be viruses. Okay? And so these are these little particles that are inside here. So when a single virus can infect a cell, and when it does, it replicates itself into many copies. And then one option then is that it kills the cell, then dies, and then these viral particles are released. Okay? And viruses come in many different types. And one of the things, though, is that they're very small. So a typical bacteria is about 1 micron across, right? And then these are much smaller, something like 50th the size of that. Okay? So they're quite small. And they were first discovered because they infect other cells, like plant cells and animal cells. All right? But they're much, much smaller. Okay? But a really interesting observation was made about them being able to actually infect bacteria as well. And it came from a very sort of strange observation in 1910, actually, concerning grasshoppers and locusts. So there was a guy named Felix Darel, and in 1910 he was in Mexico, and he noticed that all the locusts were dying off, and they were dying of diarrhea. So they had some sort of infection in their gastrointestinal system, and then they were actually dying because of this. And he found that it was a bacillus, a coccobacillus, that was in there, and that was killing the locusts. Okay? But he started growing the bacteria, and he noticed that he could make a lawn of bacteria on an agar plate, but every now and then he noticed that something was killing the bacteria. Okay, and that's something he could actually transfer it, put it into other bacteria, would kill them, he could get more of whatever this infectious agent is, and he could transfer that along. But again, they were whatever this infectious agent was, it was so small, they would pass through these filters that were normally known to stop all bacteria. Okay? And this is what this looks like. If you grow a lawn of bacteria on a plate, so normally it forms a smooth lawn that kind of looks whitish. Okay? But now what you see are what are called viral plaques. So these are places where a virus is actually infecting the bacteria, and it's killing the bacteria in those locations. So then you get a clear area where the bacteria have died. Right? So these are called bacteriophages. Okay? And so they are viruses that infect bacteria. Okay? And so they're, again, of course, remarkably small. And they look something like this. So these are the yellow things, colorized yellow here, are viral particles sitting in a bacterial cell infecting it. Okay? And this is sort of as an aside, right, an interesting idea, this poem by Jonathan Swift that, fleas have fleas that have fleas and so on, right? You get smaller and smaller and smaller things. And so one of the interesting observations that was just published a few weeks ago was that it had been thought that this bacteria, Legobacter ubiquae, was the most common organism in the world because it's a very common bacteria in ocean water, okay? And because there's this huge volume of ocean water and an enormous number of these bacteria, that this would be the single most common organism on Earth, okay? But of course, someone thought, well, but if there's something that infects this, okay, that would be even more common. And initially, no one found any bacteriophage viruses that infect them, but recently they have, and so it turns out that there's a virus which actually infects this bacteria. So now this is now thought to be the most common living thing on Earth because it infects this bacteria, which is the most common thing. But of course, there's way more viruses than there are bacteria that they're infecting. So, but I put living in quotes, okay? Does anyone know why we know? Some people think that viruses are alive, some people think viruses are not alive. Yeah, so what about it? Right, so that's one of the things, right? They need a host. It's a little bit odd, though, because, of course, there's plenty of other organisms that we don't have a problem thinking are alive, and they need a host to replicate, too. But as we'll see, the difference here is that they really have none of the machinery to copy themselves, that they need to steal pretty much everything. They're a very, very stripped-down set of instructions, okay? So they are a bit different, but nevertheless, they're a very important tool. Okay, so anyway, and then the idea is that it really can't go on to infinity, though, because nothing's been found that infects viruses, and at that point, you're down to the smallest set of instructions you could have, and obviously you can't have things smaller than single molecules that you would in any way consider alive, at least not in the way we think about it. Okay, so the last experiment here will be done. The idea was to figure out what about the virus, though, was containing instructions. So a virus would infect a bacteria, okay? And then the bacteria would basically get a new set of instructions. And the bacteria, instead of making more bacteria, would actually just make more viruses, okay? And then die. All right? People knew that. So you put a bacteria on, and the reason you got this clear plaque was that the virus changed the instructions of the metabolism of the bacteria. The bacteria stopped making the proteins it was supposed to make, okay? And instead, it made more virus particles, it died, and then those virus particles spewed out to infect more bacteria. Okay, so the question was, well, what's the molecule that's reprogramming the bacterial cell that carries these instructions? And they knew that the virus was made of just really two things. It was made of protein, it was made of DNA. Okay, so nucleic acid and protein. And so the experiment that was done, and this is a classic experiment, Alfred Hershey and Martha Chase, in 1952, is that they could radioactively label proteins versus DNA, okay? And they could label the proteins with sulfur, so remember that certain amino acids contain sulfur in them, so they could use that, but nucleic acids don't have any sulfur in them, so that wouldn't label nucleic acids. And they could use radioactive phosphorus to label DNA, and that wouldn't label the proteins, okay, at least appreciably. And so they had a way they could grow viruses with bacteria in the presence of these radioactive compounds separately. And they have a population of viruses where the proteins were radioactive with S35, and a different population where the DNA was labeled with phosphorus 32. Okay? And then what they did is they let them infect bacteria, but very quickly. So they just put the virus on for a short while. It was in a blender, and then they turned on the blender. And what would happen is that would knock the virus particles back off. Okay? But the bacteria, they could show, were infected at that point. So the virus had done its deed, and now the bacteria was committed to now making the viral particles and dying. Okay? But now what they would do is just centrifuge these bacteria and ask where the radioactivity went. Okay? So was the virus putting in protein that was reprogramming the bacteria, or was it putting in DNA that reprogrammed the bacteria? Okay? And it turned out in this case that these particular viruses, in fact, didn't put in any protein into the bacteria. They were only putting DNA in. Okay? And we'll get to examples a little more complicated where the virus puts in both DNA and protein. But in this case, this particular virus was only putting in DNA and was infecting it that way and reprogramming the cell that way with DNA. So that was the first concrete evidence that it really was DNA was the genetic information. That DNA that it was putting in, that the virus was carrying and putting in, was what was reprogramming bacterial cell to just make more virus particles. And so we'll just end with a couple other things about viruses. So this is another example of one of these T4 bacteriophage, so a virus that infects bacteria. And when it does it, it turns out that viruses can do one of two things. Okay? So it's this DNA that they inject in, so that little blue strand. And of course, the bacteria has its own normal DNA. And this viral DNA enters the cell, and actually it can do one of two things. So when we talk about it killing the cell, that's what we call the lytic phase. So this the viral DNA encodes instructions for making more viral particles and overriding everything the bacteria wants to do. And then so then the bacteria becomes filled with viral particles. They package also copies of the viral DNA, and then they escape and they can infect other cells. Okay? And they kill the bacterial cell. They can also enter what's called a lysogenic cycle. So it turns out that the DNA of the virus can actually also enter the bacterial cell and incorporate itself in, and then actually, in some cases, do no harm to the cell and sit there. Okay? And then a Q can go off, and then you'll separate out and then go back into the lytic phase and make copies of itself, reprogram the bacteria, and then kill the cell. And so, for example, you may know of like human diseases where you have a viral infection, and you can't actually ever really cure yourself because the virus will actually integrate into the DNA. And when conditions go bad, like you get sick, then the virus reemerges because the cell is unhealthy in the sense that the viral program is such that then it goes lytic again and will kill the cell. Okay? So we'll end there, and we'll pick up then with what's really in this DNA, what these instructions are. Okay, thanks.